Hello and welcome to my next video. Today I'll be doing the June 2012 F212 past paper. You seem to like the first past paper I did, so I will do another and hopefully get a few more out during the week and some chemistry videos. Right, so same format as last time. I will pause so you can do a question, then look at the answers, post how you did, any comments you want, any things you want answered. And anything that's underlined means you must have it to get the mark. Anything in brackets means you don't need it to get the mark. And any red tick that is circled means that I've gone past maximum number of marking points, but they would still be valid answers. So you have a few seconds to pause. Good. So question 1. Enzymes are important in a wide range of biological reactions. Figure 1.1 represents the mechanism of enzyme action. Name the structures represented by letters A and B. A is a substrate because it fits into an enzyme, and B is the active site. The mechanism of enzyme action was originally explained in the terms of the lock and key model. It's now more often explained in terms of the induced fit model. Suggest why the lock and key and induced fit explanations are termed models. These represent the process and show people how it works. You wouldn't really think of that, but that is what it is. Suggest why most scientists now accept the induced fit model rather than the lock and key model. More evidence and research that now supports the induced fit model. Many fish live in the Antarctic, where the water temperature can be close to 0 degrees Celsius. Scientists have studied enzymes from these Antarctic fish and also non-Antarctic fish that live in water at a temperature of 10 degrees. One of the enzymes studied has been lactate dehydrogenase, LDH, an important enzyme involved in cell metabolism. One way in which LDH works to catalyse the conversion of lactate to an important compound known as pyruvate. Scientists investigated the temperatures of reaction of LDH from Antarctic and non-Antarctic fish at a range of temperatures. Shows 3 variables that should be controlled in investigation of this type. Now, the independent variable, the one they're changing, is temperature. So the 4 main ones: temperature, enzyme concentration, substrate concentration, pH. Which one is the independent variable? Temperature, so you choose the other 3. Some suggested controls used in this investigation are listed below. Select the letter J, K, L, M or N that represents the most appropriate control to be used in this investigation. L, because you don't want a different levels of lactate, but you also want to make sure that the water temperature is constant in each area, so it is 10 degrees at one and 0 degrees at the other. The rate of conversion of lactate to pyruvate at 1 degree Celsius was found to be relatively slow when catalysed with LDH from non-Antarctic fish. Suggest reasons for this result. At 1 degree, the enzyme and substrate molecules have low kinetic energy, so there is less chance of the substrate entering the active site, so it is harder to reach the activation energy. 1 degree Celsius below the optimum temperature. It was discovered that the rate of conversion of lactate to pyruvate at 1 degree Celsius was higher if catalysed with LDH enzyme from Antarctic fish than when catalysed with LDH enzyme from non-Antarctic fish. Certain parts of the enzyme molecule from the Antarctic fish are more flexible than the equivalent parts of the molecule from the non-Antarctic fish. Suggest how a more flexible structure might help this enzyme work faster at lower temperatures. It's easy for the substrate to enter the active site. It's easy for the active site to change shape, so the induced fit model will be easier. Enzymes are proteins. The enzymes in Antarctic fish have a different structure from those found in non-Antarctic fish. Suggest how the structure of the enzymes may differ in the Antarctic and non-Antarctic fish. Different amino acids and different secondary, tertiary and quaternary structure. Suggest how the DNA of the Antarctic and non-Antarctic fish might differ. Well, if we've got different enzymes, which are proteins, they are formed from DNA when it has its primary structure of amino acids. So there must be a different base sequence, there's probably different proportions of bases, and therefore there are different genes. If species of Antarctic fish were to become extinct, their unique enzymes would be lost. Suggest why the loss of these enzymes might be undesirable. The enzyme could have future applications for us, in example medicine or industry. Suggest 2 ways in which the population of Antarctic fish could be conserved. Ban fishing, introduce quotas, protecting habitat, ex situ conservation, introducing other predatory fish to hunt. It's as I said in the last paper I did, I've left a link to the past paper, the mark scheme, so you can look at other answers because I obviously can't write them all down. So I'll give you a few seconds to do the next question. Right, good. Yet again, always find that question 2 or 3 is usually the easiest one, and this paper is very heavy on the biodiversity topic. So living organisms can be classified into 5 kingdoms based on certain key characteristics. Complete the table, and you can just see it there. So make sure that prokaryotic are heterotrophic and autotrophic. Protoctists obviously have membrane-bound organelles, sometimes have a cell wall and can be heterotrophic or autotrophic. Fungi do have membrane-bound organelles. The only purely autotrophic kingdom is plants. It must be plants, and they have cellulose cell walls, and animals have membrane-bound organelles. An unknown species is discovered. Its cell contains many nuclei scattered through the cytoplasm of thread-like structures. Suggest the kingdom this would belong to. Now, since it's got many nuclei scattered, so it's multinucleate, it must be a fungi. And classic question again. Living organisms can also be classified into 3 groups called domains. Outline the features of this system of classification compared with the 5 kingdom system. Prokaryotes split into eubacteria and archaeobacteria. These reflect differences such as rRNA, DNA replication, and DNA and RNA is normal. All eukaryotics grouped together because of similarities between all eukaryotics, such as a nucleus and chromosomes, membrane-bound organelles, linear DNA, stuff like that. So I'll let you pause. Good. Bats are the only mammals that can truly fly. Many species of the bat hunt flying insects at night. Bats are able to use sound waves, echolocation, in order to help them find prey in the dark. Suggests how the ability to use echolocation may have evolved from an ancestor that did not have that ability. So we're looking at evolution. What's the mechanism behind evolution? Natural selection. Now you get 1 mark for same mutation, 1 mark for random. So say both together. A random mutation occurs due to the selection pressure of competition for food. The individuals with the mutation survive and pass on their genes over many generations. The pipistrelle is the most common species of bat in Europe. It was originally thought that all pipistrelles belonged to the same species, Pipistrellus pipistrellus. However, in the 1990s it was decided that they would split. There were 2 species, the common pipistrelle, Pipistrellus pipistrellus, and the soprano pipistrelle, Pipistrellus pygmaeus. God, so many words. And there's a nice table. Name the genus to which the soprano pipistrelle belongs. Well, they both belong to the same genus, Pipistrellus, capital P, underlined. Using the data in table 3.1 suggests why pipistrelles were originally classified as 1 species. They've got the same body mass, similar wingspan, similar colour. State 2 pieces of molecular evidence that can be used to identify organisms as belonging to different species. Genetics, RNA, amino acid sequence, cytochrome C. Describe how it is possible to confirm over a longer period of time whether 2 organisms belong to the same species or different species. When 2 organisms interbreed, if they produce fertile offspring, then they are the same species. That's basically asking for the definition of a species. And the opposite, if 2 organisms interbreed and produce infertile offspring, then they are different species. And then the first of the 2 big quality of written communication questions. The soprano pipistrelle has an echolocation call that is high-pitched. The common pipistrelle has an echolocation call that is low-pitched. Variation within and between species can be as a result of genetic or environmental factors. Whatever the cause of variation, the type of variation displayed can occur in 2 different forms. Using the pipistrelle as an example, describe the key features of both forms of variation. In your answer, you should make it clear how genes and environment refer to each form of variation. That means once you need to talk about how genes in the environment affect one type of variation. So the two types are continuous variation. This is caused by many genes in the environment. Quality of written communication mark there. There is a range of values, so many intermediates, e.g. body mass and wingspan within a species. There's discontinuous variation. This is caused by one or a few genes. There is no or little environmental effect. So if you've got green eyes, you're not going to change that genetically. There are discrete groups with no intermediates, example gender. And I'll let you pause now for the next question. Good. Figure 4.1 shows a representation of part of a carbohydrate molecule called agarose. One of the subunits of agarose is a sugar called galactose. Identify the type of carbohydrate molecule which the carbohydrate agarose is an example. It's a polysaccharide. Starch contains a carbohydrate called amylose. Amylose does not contain galactose. Using the information in figure 4.1, identify one similarity and one further difference in the structure between agarose and amylose. They're both unbranched, they both have a chain, they contain only carbon, hydrogen and oxygen. Differences: different types of glycosidic bonds, one is a 1,6, while the other is 1,4, different sugars and different monomers present. Agarose forms part of a more complex carbohydrate called agar. This is used as a growth medium for bacteria. Bacteria cannot break down the agarose in agar. Suggest why bacteria cannot break down agarose. They do not have the correct enzyme that has an active site that is complementary to agarose. A student wished to demonstrate experimentally that bacteria cannot break down agarose. The student used a culture of Escherichia coli, E. coli, bacteria which had been growing in a solution containing starch. Two tubes A and B were set up as follows. Tube A, actually you can read all that. Anyway, explain the purpose of tube B, which has no reducing sugar present. It's a control to compare with tube A to show that it was the bacteria doing it and not some other force. The student wrote the following conclusion. My experiment showed that bacteria must be able to break down agarose. This is because reducing sugar was present in tube A. Suggest an alternative explanation for the presence of reducing sugar in tube A that is not consistent with the student's conclusion. There might have been some starch present in nutrient solution at the start. There might be a different bacteria in there. You can't control it, so just one bacteria. Suggest two ways in which the reliability of the experiment could be improved: repeating and sampling each tube twice. The student did not have access to a colorimeter when testing solutions for the presence of reducing sugar. Describe how the student could carry out a chemical test for reducing sugar and suggest how they could estimate the amount of reducing sugar in the sample from tube A. So Benedict solution at 80 degrees, a precipitate will form that changes colour from blue to green to yellow to orange to brown to brick red. The further colour change, the greater the concentration. That's simply put. Another student suggested that agarose may have been broken down to a non-reducing sugar. Describe how the test for reducing sugar could be modified. Add hydrochloric acid, then add sodium hydrocarbonate, and then carry out the Benedict's test again and look for a positive result. So I'll let you pause there for the next question. Good. An investigation was carried out into the effect on lung function on giving up smoking. The investigation measured the maximum volume of air that could be exhaled in 1 second in a group of people who had stopped smoking and in a similar group of people who continued to smoke over a 5-year period. Using the information in figure 5.1, calculate the percentage decrease in FEV over the 5-year period of people who continued to smoke. So that's the circles. You look for the change in the FEV and divide that by the original FEV times by 100 to get a percentage. Remember, it says 1 decimal place, so 11.3. Here's the good old describe the trends question. Smokers FEV lower than non-smokers. Smokers FEV decreases, 1 mark, from and then 2.75 dm cubed at 0 years to 2.44 dm cubed at 5 years. Comparison mark. Non-smokers' FEV increases and then decreases. And the other quality of written communication mark. One of the symptoms of smoking is the development of a smoker's cough. Explain how smoking causes a smoker's cough and how the cough itself can lead to further problems in the lungs over a longer period of time. In your answer, you should clearly distinguish between the development of the cough and the effects of prolonged coughing. So you need one cause, one effect. So it's caused by tar. Tar destroys the cilia and stimulates the goblet cells to release more mucus, which builds up in the airways and cannot be removed, meaning more bacteria, viruses, pathogens get caught in the mucus. The cough is an attempt to remove microbes to get more airflow going by removing the mucus. Now the cough can damage the airways, particularly the alveoli and elastic tissue, forming scar tissue on the walls, which thickens the airway walls, reducing lumen size, restricting airflow. It can also, the more diseases can lead you can make you prone to infection. Also, elastic tissue can be destroyed, which means you get less lung capacity. Chronic obstructive pulmonary disorder, COPD, is a combination of diseases that can result in coughing, breathing difficulties and fatigue. Name 2 specific diseases that contribute to COPD. Emphysema, chronic bronchitis, and the other is asthma. One form of COPD develops because enzymes are released by phagocytes entering the alveoli. The enzyme action can break down elastin in the lining of the bronchioles and alveoli. Using the example of elastin breakdown to explain the induced fit hypothesis of enzyme action. So this it's not asking you how it's broken down, it's asking you to talk about the induced fit hypothesis. So elastin is the substrate. Elastin binds to the active site, which changes shape to create a closer fit, where more bonds form. This creates an enzyme substrate complex. So I think that was probably quite a hard question. There's a lot of knowledge in there to get all those marks. The whole question. So I'll let you pause for the next one. Good. This is a very weird start to a question, I think. The traditional English folk song, the Derby Ram, or Derby Ram probably, contains the lyrics, "As I went out to Derby all on the market day, I spied the biggest ram, sir, that ever was fed on hay." I don't know why that was there, but you get a nice song. Yay. The song is likely to have been inspired by the successes of farmers in the 18th century who developed a sheep known as the Dishley ram. The ram gave rise to a breed which grew more quickly, producing more wool and meat than other varieties of sheep. Suggest how it would be possible for farmers in the 18th century to produce a larger, more profitable variety of sheep. Now, like the one about the evolving of the echolocation, you just get one mark for stating the mechanism, selective breeding. Select sheep, and you explain it. Select sheep with the desired characteristics and breed them together. Then select the best offspring, do this over many generations. Since the 18th century, other ways of improving productivity in sheep have been developed. State one further way of improving productivity. There are many, I just chose antibiotics. Crop yield can be improved by the use of fertilizers in the 18th century. These are likely to have been organic fertilizers in the form of manure or compost. Shows how organic fertilizers improve the yield of plant crops. Compost is broken down by decomposers, add minerals and ions to the soil. That's nitrogen, phosphorus and potassium. Lack of minerals is a limiting factor. Inorganic fertilizers are not directly toxic to living organisms. However, the excessive use of these fertilizers can lead to a reduction in the biodiversity of farmland. Suggest how the excessive use of inorganic fertilizers on farmland can cause a reduction in biodiversity. The fertilizers promote growth of one species of plant, the crop. This plant outcompetes other plants and disrupts the food chain. Explain why a reduction in biodiversity may present problems in agriculture in the future. Loss of genetic variation and useful genes, example the gene for pest resistance. There is also a loss of pollinators and pest predators. So one more question to go, very 6-mark question next, so pause and you can have a go. Good. Right. Select the most appropriate term from the list below to complete the table. Many words here. So definition, sampling which the observer does not decide when and where to take measurements. Random sampling. A representative group of organisms that are selected from a population. Sample. An area in which an organism lives. Habitat. a measure of the relative number of individuals in each species, that's species evenness, the frequency of occurrence of plants in a particular area, that's abundance of plants, and the number of species present in a particular area is species richness. And that's the paper done. I hope that helps. Yet again, it takes almost exactly 20 minutes to do these. Cool. So I hope that helped and you enjoyed. Any questions, ask. Please comment what marks you're getting, what sections you're good at, and if any help you want. And you can email me, like, comment, subscribe, whatever you want. So yeah, thanks for listening and goodbye.